Okay, we'll start. Hello everyone and welcome to MDC Connects. I'm Sarah Brockbank. I'm a lead scientist at Medicines Discovery Catapult and I'm hosting the webinar today. I'm here with my colleague Darren Holmes, who's behind the scenes looking after Zoom for us and keeping us connected. So this year MDC Connects is focusing on complex medicines. And we've been joined for this series by a whole host of experts from across the UK. And the experts have taken us step by step from concept through to the clinic. And this week we are on webinar number 5. And if you have not been able to join us for any of the previous webinars, these are available on the MDC website as recordings. But for today, we are understanding safety and efficacy. So in this session, we're looking at how to determine preclinical safety and efficacy in vivo and how complex medicines compare with conventional drug discovery. And I'd like to welcome 3 speakers. Firstly, Jenny Worthington, who's the co-founder and director of science at Access Bio. And Jenny's going to discuss how to plan an in vivo efficacy study for a complex medicine. Then we're going to be joined by Juliana Maynard, who's also from here at the Medicines Discovery Catapult, where she heads up our preclinical imaging unit. And Juliana's going to use case studies to describe how imaging technologies can be used to determine that the medicine has reached the target tissue. And finally, Richard Knight, who's director and co-founder of Connects, and Richard's going to address the question of safety. Which may be the same question for conventional medicine, but perhaps more involved to answer that question. So we will take questions at the end of each presentation. And if you wish to ask a question, if you can type that into your Q&A box at the bottom of your screen. And it'd be really helpful if you can type these in as you think of them, so they're ready for me to ask when the speaker finishes talking. So without further ado, over to you, Jenny.
Thank you, Sarah. Let me try and share my screen. There we go. Okay, so first of all, I'd like to thank Sarah and her team for actually inviting me back to give another talk this year. Last year's must have been okay to get another invite. So thank you, Sarah, for that. And then secondly, I'd like to thank each of you for giving up your time to come along and hear me witter on about the type of work that we do at Access Bio. And I'm going to give you quite a higher level talk. I'll not go into too many specifics, but just give you an idea of some of the things that you might need to think about and consider when you are thinking about taking your complex medicines into in vivo studies. My fellow presenters, Juliana and Richard, then will delve more deeply into PK and safety of these medicines. So for those of you who don't know me, my name is Jenny Worthington and I am the co-founder and director at Access Bio. I started my career in cancer biology at university. So I've been there and had that academic experience of being a postdoc, moving up through lecture and senior lecture before I decided then to branch out into industry. As a CRO, Access has seen many different types of medicines, and what I want to do today is to give you our perspectives on complex medicines and maybe try and identify if we should think about these in a different way than conventional molecules. So today, my talk, I'm going to give a very brief background to Access Bio and then have a look at the steps that are involved in preclinical analysis of these complex medicines. And as I say, kind of try and identify if these are different or not from the steps that would be involved in standard molecules. Look at these steps then individually in a bit more detail, and then finally round up with a few bottom lines in this. And I think if you take away nothing, so if you stop listening to my talk now, I think the one thing that I want you to take away from today is to be humble and get experts in the field involved as quickly as possible. So talk to people who know about PK, talk to people who know about safety, and get them to help you design your studies so that you're collecting the correct samples and looking for the correct things as you go along. So then this is obviously not a marketing presentation, but Sarah did say we could put one slide in about the company. So this is it from Access Bio. So we're a CRO and we work mainly in oncology and inflammation. And alongside that in-life side of the work, we also have cell biology experience, so looking at things like immunohistochemistry and FACS analysis, along with all your standard cell biology techniques, Westerns, ELISAs, molecular biology. What I want to emphasize is that as a CRO, we deal with clients from lots and lots of different backgrounds. So we deal with large pharma through to two-person biotechs and everything in between. We deal with lots and lots of universities in the UK and in Europe. So we have a lot of experience with different clients. We know that each type of client has a slightly different outlook on things. And I would say, please, please, please don't be shy about approaching us and asking questions and trying to get advice into seeing how we can help move your program onwards. So Sarah, that's it, marketing bit over and done with for now. So we go on to the more important part of the talk. And what I've done here is it's a very simple look at how we think about moving medicine into preclinical testing, and this is a very simplified version of this, and certainly the arrow just doesn't go in that direction. It moves backwards and forwards between them all. PK, pharmacokinetics. We need to know about the dosing route that you're potentially going to use. We need to know about exposure levels, both in general and in the target tissue, before you can even consider taking it forward. If you've got this wonderful new complex medicine, but when you inject it into animals, there's no exposure anywhere, or it just disappears as soon as you put it in, there's no point moving it forwards. To move towards an efficacy study, we need to know about the linearity or not of dosing. If we give a higher dose, does that result in greater exposure or not? Biodistribution, when you put this into the body, where does it go? When we're dealing with a small molecule, we know that it goes throughout the body. Is that the same with your complex medicine? And that's because complex medicines are so complex and so diverse, you'll hear me just talking about your complex medicine because there are so many different types. It's difficult to deal with them specifically. But does your medicine accumulate somewhere specific? Have you got a targeting approach to your work? So these are questions that are all going to feed then into an efficacy study, which is the bottom line. Does your medicine have any efficacy? Does it work? Is there any point in taking it through all these next steps? Generally, an efficacy study will involve multiple doses. Do we see any adverse effects with these? So data from your efficacy study and your biodistribution studies can ultimately then feed into toxicity studies. As I said, things don't go in a straight line, but you do tend to go backwards and forwards as you gain more information and check on other issues that you generate. But the bottom line is essentially there is no difference in this process if you're looking at complex medicines. However, as with all science, there are very subtle differences that need to be considered. And as I mentioned, Juliana will deal with PK in much more detail. But I just wanted to highlight a couple of things that can go wrong and a few things to maybe have a think about early on. So to get to this nice PK profile of drug A, there's actually quite a few steps that you will have to think about before you can actually get a successful type of PK. So circulation, I've mentioned. In the body, due to the very nature of it, there are going to be areas where you get differential blood flow rates. So if you have an area of very fast flow, is that flow rate going to have an effect on your complex medicine? Is it going to burst it apart before it even gets to where it needs to go? Is that going to result in massive exposure in the general body that you don't want to see? What about the cells that it encounters within the bloodstream? Are these going to have an effect? Are these going to mop up your medicine? Stability. How stable is your complex medicine? And actually, this is, as well as being important from an in vivo point of view, this is actually really important for collecting samples. So, for example, how does it react at body temperature? How does it react on ice? If we're collecting blood samples, is there a time scale involved with that? How does it react even in the freezer? So these seem like basic questions to ask, but they do need to be considered. Clearance, does this occur via the normal situation in terms of liver and kidney, or is your medicine mopped up by the macrophages and other phagocytes within the blood supply? Does this accumulate in organs and cause toxicity? So there's lots of things to think about in terms of PK. One thing I will highlight is that your bioanalysis method may not be simple and straightforward. In fact, it's very likely it will not be simple and straightforward. If I give you an example, if we're dealing with small molecules and we're sending them away for bioanalysis, there will be minimal method development required for that. We use a standard approach, and the method development is about £500. With your complex medicines, it's going to cost a lot more than that. And at the start of your program, you need to factor in a good chunk of money for method development for your bioanalysis method. In terms of actual collecting of the samples, might not necessarily be a simple approach. The method development process will highlight what needs to be done to the samples after they're taken. Bottom line, get people who know how to deal with these things on board quickly to advise your study and advise you on the type of samples that you should be taking. If we then move into biodistribution, what happens to your complex medicine when it's introduced into the body? I've broken this down into considerations just in general throughout the body, then thinking about the target organ, and then within cells. And I will apologise but not apologise. So when I talk about target organ here, generally I'm talking about tumours, because that's the cancer approach, the oncology approach is the one that we internally have had most experience with. So in terms of biodistribution, then things to be thinking about, is there a difference in the biodistribution if you administer IP, IV, orally? You know, I guess we've had most experience with the complex medicines with IV dosing rather than anything else. Then you're thinking about extravasation. Does it actually get out of the blood supply? You're thinking about targeting of organs. Do you have some kind of targeting moiety on your molecule to target, for example, tumour or whatever? And then you're thinking about physical barriers, the tight junctions that are seen in many organs within the body. Then if we're thinking again, moving down into that target organ, so tumour vasculature is crap, quite simply. You know, you can have a wonderful medicine and you get it to the tumour, but it doesn't actually get all around the tumour because the vasculature within that tumour is so heterogeneous that provides a massive issue. Connected to that then is the interstitial fluid, which can actually stop your molecule from getting into the tissue. The ECM concentration again can affect that. And then a very simple thing that's not necessarily to do with tumours but other organs. Have you got mucus that your molecule has to get through to have its effect, and does that make a difference? Then actually some of your complex medicines might need to actually enter the cell to work. So what about cells? Are the cells able to take up your complex medicine easily? And then actually, that's apologies, that actually says cell heterogeneity on the bottom of that. You know, we're thinking about tumour cells. We have a whole range of different types of tumour cells that will be resistant potentially to different types of medicines. So given, as I've said here, given the target and the proposed mechanism of action of your medicine, your issues that you have to think about with biodistribution or opportunities will be project specific. So in terms of what Axis does, we tend to deal with a more general approach to biodistribution. And I wanted to just take this opportunity to share a couple of different approaches that we've taken to look at tracking therapeutic agents throughout the body. So then in this first gel, the product actually had some nucleic acid elements within it. So we were able to design primers that were specific to that sequence and use these to just do a very simple yes/no to whether the product could be detected in specific organs. So lanes in that image, lanes 2 to 16 are different organs, and you can see that the product was fairly well distributed throughout that body and that there's evidence of accumulation in certain organs. So the second example shows a fluorescently labelled set of products. Now they were given at 2 different doses, and so the 3 rows that you see are 3 different products, and obviously then the columns are the 2 different concentrations. So just to highlight, anything that red contains more of the fluorescent agent. So these images were taken 24 hours after dosing, and you can see that in the top left panel that at the lower dose the product's mainly found in the liver, but at the higher dose it's obviously everywhere. So we don't have very much targeting there. Middle row, we're seeing something similar, but there's generally less product there. The bottom row is actually the most interesting. At the lower dose, we actually don't see very much product left there after 24 hours. But at the higher dose, you can actually see the red signal is focused, and I can tell you that those red areas are tumours. So that product was able to reasonably successfully target tumour specifically. So then moving on to efficacy studies, and I will say up front, do not expect that your first efficacy study will be the last one, because you will need more. You will run into problems. But the bottom line with efficacy studies is that for complex medicine, there is no difference in planning an efficacy study compared to a normal small molecule, for example. You still want to consider your best model system. You still want to consider adverse effects. You still want to think about how many dosing occasions you're going to get, and then tissue collection. What tissues are you going to collect at the end? One of the very important things that I would suggest that you consider is that you generate sufficient material to allow you to carry out these in vivo studies. If you can afford it, think of what you might need and then double it, because you will not, the first study will not work perfectly. There will be at least another iteration of in vivo studies there.
Start wrapping up, Jenny.
Yep, I will. Yep, almost last slide. So complex medicines. Does this mean complex toxicology? And I'm going to be very honest, I don't know. That's why we have Richard on to talk about this in more detail. But what I would say is that your efficacy studies, you will generally have multiple dosing. So collect as many tissues as you can from those studies. Get your toxicology expertise on board as early as possible, and they can advise you on what you should be looking for and what samples you need to take. This is just a summary slide that I don't need to go through. It's got everything on there that I've already discussed. As I said, bottom line, get experts who have done this before on board to help guide your study. Thank you.
Okay, thanks, Jenny. That's great. In the interest of time, I'm going to press on, but if you do have questions for Jenny, please put them into the Q&A box, because Jenny will be able to see those and answer directly to you. And if we do get any time later on, Jenny will come back to you. And
yep, that's fine.
Okay, Juliana, do you want to?
Fantastic. Yep, I'll just try and start screen sharing. Okay. Hopefully that should be coming any second. Yeah, I'll just go back to the beginning. Sorry, a snippet of the main part. So thanks, Jenny, for your really excellent presentation, and thanks everyone for giving me the opportunity to speak today. So as Sarah said, my name's Juliana Maynard. I'm from the Medicines Discovery Catapult, and we're based here in Alderley Park. So today I'm going to talk about some of the work that we've been doing in the complex medicine space, and specifically how we've been working on establishing the physiochemical characterization of a complex medicine. And I've included some case studies of actual projects that we're really delighted to be working on, and how we're helping our collaborators to do just this. So I guess, what are the opportunities and challenges then within this space? And Jenny has articulated this brilliantly. So the complex medicines area and arena are increasing rapidly, and they do offer absolutely huge opportunity to be transformational in drug discovery. And nowadays, biologics and complex drugs make up a significant proportion of those products that are actually currently in development. But there are challenges, however, and that is in associating and determining the pharmaceutical bioequivalence of these drugs compared to something that you would do with a conventional small molecule. So characterization is absolutely key, and especially in getting that preclinical candidate to a successful marketing authorization, new and novel approaches are needed to understand those pharmacokinetics and characterization. So establishing that relationship between what you're seeing preclinically and its in vivo performance will significantly increase the likelihood of clinical success. And so what can be done, and how can we help and provide access and validation of both in vitro assays and in vivo animal models? And I know my colleague spoke earlier in the series about the in vitro assays we're doing here in the Catapult, so I'm focusing on the in vivo side today, but very much the absolute sort of cascade of a complex medicine. When it goes into the body, it's circulating in the body, it will accumulate, it will penetrate into the tissue that it needs to go to, it will stay there, it will internalize, and it needs to then release its cargo. And by being able to sort of track and characterize that form right through from the systemic level all the way through to the cellular level becomes essential in characterization. And imaging offers a huge potential for that characterization in a number of ways. And we have a number of capabilities here in MDC, and we're well set up to provide this information with examples of use in both, as I say, in vitro and in vivo model characterization. So today I'm just going to focus on those in vivo case studies and the facilities that we have within the Catapult. So we do have a broad and unique offering of imaging capabilities and expertise in multiparametric imaging modalities. So we have access to positron emission tomography, computed tomography, high frequency ultrasound, our bioluminescence and fluorescence imaging capability. We also have a lot of ex vivo imaging, for example, mass spectrometry imaging, our digital spatial profiler, and advanced microscopy as well. So we're very much across all of the areas. And in terms of accessing those tissues and models, so we have our own home office project license, we have our radiological license, and we have access to a wide range of animal models across therapeutic areas and also naive animals themselves. We have our own novel radiochemistry, so we have our ability to be able to actually radiolabel those complex medicines. And so we really think that there's an opportunity in that multimodal enhanced interpretation of functional and molecular data. So today I've just thrown and chosen 3 case studies to talk through and the companies that we're currently working on. And as Jenny led into, we're just going to focus on what we're really looking at in that pharmacokinetics and that accumulation and distribution of a complex medicine. So drug distribution itself, which represents the extent to which it's present in the extravascular tissues, and the rate and degree by which it's distributed will depend on a number of factors. So it will depend on blood flow, capillary permeability, protein binding, and accumulation and redistribution to other sites. So in the complex medicine space, understanding that distribution is absolutely key. And the 3 companies I've chosen today that we've been working with are zeri Healthcare, Sixfold Bioscience, and the University of Leeds Therapeutic Microbubble project. So in this first example from zeri Healthcare, they are developing a nanomedicine that has significant promise to transform radiotherapy. So 50% of cancer patients receive radiotherapy, and a sufficient dose will indeed actually kill any cancer cell. But that dose is limited by toxicity to the surrounding tissues, and even small increases in dose will actually give a significant improvement in patient outcome. So zerion's product offers the opportunity to give substantially increased X-ray dose enhancement, but it won't damage healthy tissue. So it's a really exciting opportunity to really be able to transform radiotherapy. So what does darion have? They are titanium oxide nanoparticles, and they have many fantastic properties. They're a good size for actually having and receiving cellular uptake. They strongly absorb the X-rays, and they're effective in hypoxic tumours, which are obviously renowned to being resistant to both chemo but also radiotherapy. So Zeri are currently in preclinical testing. They have two disease areas that they're really focusing on at the moment. So their nanoparticles are directed and injected directly into the tumour prior to radiotherapy. As I say, their targets are head and neck cancer and also pancreatic cancer, which is notoriously renowned for having a very low survival, 5-year survival rate. So real fantastic opportunity to make wonderful steps in this area. And the question that Darion raised to us was actually, how can we determine the distribution and characteristics of the nanoparticles? So when they're injected into the tumour, are they distributing in a heterogeneous manner, or where are they going to? So we have been using two of our in vivo capabilities here in MDC to do this. The first being our high frequency ultrasound. So here we've actually got the needle, you can see the needle here, and we're injecting this into the actual tumour. So if I play my video, you can see the nanoparticles as they go into the tumour themselves and where they're dispersed to. And what we've been able to see is that map that signal in real time as well. So the graph on the right is that signal intensity and that echogenicity as those therapeutics go into the actual tumour. This next slide is just seeing this in 3D, so we can quantify that tumour volume in a 3-dimensional way. On the outside, we can just delineate and see the actual skin of the tumour. It's a subcutaneous tumour, and we can see that nanoparticle distribution within the tumour itself in yellow on the right. We've also then taken the tumours out and we've looked using micro-CT ex vivo assessment of actually what we're seeing in the xenograft as well. And so what we're seeing here, oh, sorry, just play my video, is you can see in 3 dimensions as these tumours are flowing around exactly where those nanoparticles are going to. And what we're doing now is we're really working with Darion and other collaborators in how we can optimize that nanoparticle distribution to ensure that they're going throughout the actual tumour. So in the interest of time, then if I just move on to our second collaborator, so this is with Sixfold Bioscience. Here they're looking at delivering RNA therapies to treat disease. So I think over the past decade, the development of RNA therapies and specifically siRNAs has been accelerating. And compared to both small molecules, but also more complex antibody-based drugs, they can act on an unrestricted choice of therapeutic targets. But they are inherently unstable in the blood. And there's an urgent need for those delivery platforms that can really overcome those shortcomings to take these into clinical translation. So Sixfold have developed a really exciting programmable oligonucleotide delivery system, which we're known as pods. And this will actually be a delivery platform to take the RNA and deliver this to specific cancer cells. So the pods, as I say, that they're known as, they consist of a non-toxic RNA scaffold, which will carry that therapeutic cargo to the tumour. It has a targeting ligand, so it ensures that only disease cells will receive the actual pod itself. And as I say, they have their therapeutic targeted design attached. And then their modular design means that we can actually attach multiple functionalities to these. So the actual platform and system itself, again, offers huge potential to work across targeting areas. So the siRNAs have, as I say, potential to improve treatments in a huge amount of indications, and Sixfold are currently working and really assessing their priority and really where they want to target first. But the 2 approved siRNAs that are currently approved are actually for the treatment of rare liver disorders. And we know that this market is going to grow rapidly throughout the years. So what were we really looking forward to do with Sixfold to move them towards clinical translation? So one of the key questions was, how can we determine the proof of the distribution and where their pods were going to, what should the organ of interest they be looking at, but also proof of concept for their delivery. So looking at efficacy and alternative platforms and therapeutics. So here, just to go into some of the first initial studies that we've been doing. So we've chosen two techniques to look at here. The first being the near infrared imaging. So here what we've got is actually we've labelled the pods with a size 7 dye. These have been injected into the mouse. These were actually injected intratumorally initially just to see what that signal was of the size 7 dye, and looking at where the longitudinal signal would be in vivo. We also took these pods out ex vivo, and we looked at them under microscopy. And what we can see here is the little red is the actual size 7 pods themselves, which we were able to determine with microscopy as well. So trying to really tease apart which cells they're going to and get that characterization. So we're really excited about the size 7 labeling, but we are now moving into zirconium pod labeled PK assessment. And what we want to do here is get really micro zirconium labeled blood samples at very, very early time points, put them on a gamma counter and really be able to map out that PK/PD characterization for 6-fold bioscience. And I think because of the sensitivity of the PET system, then we will move forward with a lot more zirconium pod labeling for the in vivo studies. But what we want to do is also then look at for 6-fold the proof of concept and efficacy of delivery. So are we shrinking the tumours? But if we're shrinking the tumours, are we doing so and are we able to do this to a superior standard of care, so an alternative siRNA, but also using a different platform? So are the pods going to be superior to other platforms such as liposomes that are in way? So we're doing multiple distribution and efficacy studies with 6-fold, and we're really excited about our long-term collaboration together and really moving forward this exciting technology with them. So my final case study for today is one with the University of Leeds, and one we've been working on for several years. We're really excited about their Therapeutic Microbubble Consortium. So I know Louise presented brilliantly on this last week. So today I'm just going to go through some of the actual imaging components of the work that we did to help, which is very complementary to the efficacy work that they did. So as a refresher to the Therapeutic Microbubble platform that Leeds and Louise mentioned, so most cancer patients, as we all know, do receive chemotherapy, but the sufficient side effect is the dose, and it would kill any cancer cells. But as I say, delivering that therapeutic dose without side effects has definitely got its challenges. And there's been so much interest then in that nanoformulation and those targeted delivery platforms. So the University of Leeds have developed a therapeutic, VEGFR2 targeted, so they're using that targeting approach, and an ultrasound triggered delivery method that will really improve the therapeutic range of cytotoxic drugs. So just to give a reminder of what they are itself and what they can do with the ultrasound providing cavitation of the cell. As I say, they're tiny bubbles of gas, and when they're used with ultrasound imaging, they produce increased contrast. So we can image that increased echogenicity on an ultrasound scanner. So we have the VEGFR2 targeting moiety, so it will go to the actual tumour. It has its drug payload inside it. And as I say, that ultrasound trigger directly onto the tumour will then release the drug, causing cavitation of the cell and the drug to go inside of it. And what we've been doing with MDC, again, in a similar way to sort of 6-fold, is understanding how we can really help to characterize and provide that proof of concept of the therapeutic microbubble platform and move it towards clinical studies in man, which we're now focusing on, which is really exciting. So as I say, what we did is we actually did some PET imaging. So another technique that we use widely within the Catapult. And here we actually labelled the liposomes. So we put a chelator inside the liposomes, and we were able to label the microbubbles themselves, put them into the body and really see that distribution. So what we were able to see, if we look at the graphs on the right-hand side, is that actually we do see improved tumour accumulation of the zirconium-labelled therapeutic microbubbles and the targeting agents compared to if you just had labelled liposomes alone. So those therapeutic microbubbles are providing an additionality and getting that drug to the tumour. I think the images speak for themselves, and that there's a lot of liver and spleen uptake, which was astonishing. And that clearance and distribution is as you'd expect in a liposomal method, that we are clearing through the reticuloendothelial system. But we can show, and obviously in combination with the imaging studies themselves that University of Leeds have done, that we can enhance that efficacy of cytotoxic drugs in vivo by increasing the concentration of drug into the circulation. As I say, we're looking at that across time. So this project as a whole was really showing that compelling evidence, validating the mechanism of action and potential of the platform. And we've recently published this data in Theranostics last year. And as I say, now we'll move into man, which will be a really exciting outcome for this project. So just to really summarize then, I guess what I've chatted through today, and hope you found those examples interesting, that in vivo imaging offers huge potential as a platform for non-invasively measuring those pharmacokinetics. And very much alluding to what Jenny said, each one's very different, and it's exciting to work on each and every project to be able to understand which imaging modality that we can choose that is the best for them. And we're, as I say, we're very excited to have such a wonderful range of sensitive techniques in-house. And yeah, just really delighted and excited to be working with our collaborators on these and future collaborators as we come. So I think that exploitation of our current capabilities, but also developing those emerging opportunities, understanding new techniques, will just really push these complex medicines faster into clinical development. I think that's my final slide for today, then, just to say a huge thank you to all of my colleagues within the Medicine Discovery Catapult, but also very importantly our collaborators and allowing us to work with them. Really excited about working with them all, and thank you to you all for listening to me today, and I hope that was useful.
Thanks, Juliana, brilliant. Okay, and again, in the interest of time, I'm going to move on. I'm absolutely brimming with questions for both you and Jenny. If time permits at the end, we might take questions for all three speakers. If not, I think we'll just press on with Richard now. Thanks, Richard, if you want to put your slides up.
Can you see that, Sarah?
Yeah, brilliant.
Great. Well, thanks everybody. My name is Richard Knight from Connects, which is a preclinical safety group also based at Alderley Park. First of all, I'd like to say thanks to Sarah and thank you to the MDC for inviting me to talk about the safety evaluation of complex medicines and whether they present a different challenge from a safety perspective. I guess to cut to the chase, and I think Jenny's touched on this already, I mean, I think in principle the answer is no, it doesn't. And I think we should take the same considered scientific approach, whatever the therapeutic modality. And it's about understanding the biological implications, characterizing the hazards, and then assessing the risk to patients. It just becomes a little bit more complicated the more complex the test item and the more components there are to consider. Now, safety evaluation is obviously a well-regulated environment, and there are a large number of regulatory guidelines. Organizations like ICH and other national and international bodies produce guideline documents for non-clinical safety studies to support clinical trials. It's the ICH M3 guideline that is the main one to be referred to. But there are also a whole host of other safety-specific guidelines, for example, S6 on the right-hand side there if you're working with biotechnology products, or S9 for anti-cancer agents. But it is important to remember, I think, that these are not rules, they are guidelines, and they're there to reflect the agency's thinking on key topics, things like carcinogenicity or genotoxicity or immunotoxicity testing. So they're not comprehensive, and there's often no specific guidelines on some of the more complex approaches. So it's up to the sponsor to decide which aspects of these guidances that they consider relevant to their product. So I think in essence, you know, everybody's required to go back to first principles and think about the risks that are raised by the novel agent. What data would be helpful to put that risk in context of the clinical trial? And I think, you know, as part of last year's MDC talks, I introduced this concept of the target, the chemistry, and the patient as the framework with which to begin this exercise. And it's a way of identifying the key risks for any program, really. And I think we can build on that as well for complex medicines. If we kind of work through that a little bit with some relatively simple molecules, I think whatever the platform, the intent is to modulate the therapeutic target or therapeutic process. So it's important to understand that, whatever the therapeutic approach. And we know that something like 50% of all clinical toxicities are due to on-target pharmacology. So it's important to understand that as well as we can, as well as how that varies across preclinical species. And for small molecules, there are further questions that are raised by the chemistry. So how selective is it? Does the chemistry bring in secondary pharmacological activity at unintended targets that might be important? Is the chemistry genotoxic? What are the ADME properties? So are there safety implications from metabolites by the route of excretion? Are there impurities to consider, for example? And then the patient context is always very important to understand. There's no point in developing a novel treatment for a disease if the patient can't then take it. And so you can contrast that focus then with that of monoclonal antibodies, for example, where the exquisite specificity for the intended therapeutic target sequence and the biological nature means that some of these off-target considerations aren't really applicable. So things like secondary pharma profiling, hERG activity, genotoxic assessment when you've got no potential of interacting with DNA, these kind of fall by the wayside as not really relevant, really. But as you get more complicated, these things come back into the fore. So if you have bispecifics, for example, or other novel constructs, then they might well become more relevant. For ASOs, these kind of fall somewhere in between. So that, again, there's no specific guidance for an antisense oligonucleotide. And so you have to bring in aspects of both small molecule and biologics thinking. So they do tend to be highly selective for the intended mRNA target, but there may be potential for sequence overlap. And so that's important to understand. We know there are various platform-related toxicities with ASOs that we need to be aware of. And unless we're introducing novel chemical modifications to the usual backbone, then there may be less concern about metabolites and specific impurities, and they don't tend to interact with DNA, for example. But then as you introduce further complexity, I guess these additional components all then need further consideration, and you can work through the same process and thereby construct a safety package for your IND or your CTA with reference to the guideline, but tailored to the specific needs of your complex medicines, and understand whether, given the variety of things that fall under the umbrella of a complex medicines, which of these kind of considerations become relevant. So it's really focused on asking the right questions in the context of the safety considerations of the patients. So often the purpose of creating and developing these complex medicines is focused on deriving a more effective targeting of a therapeutic to tissues and to try and improve the therapeutic index. So I've got three short examples to share today. The first one is an AstraZeneca program. So AstraZeneca had an Aurora B kinase inhibitor called AZD1152 or barasertib. And you can see from the Kaplan-Meier plot on the right-hand side there that this is a compound that did deliver clinical benefit in AML patients compared with standard of care. But there was a but here, and the but was that it had to be delivered as a 7-day continuous IV infusion. So that really restricted the application of this compound. So it was limited to hospitalized patients. Many patients were not suitable to that kind of a dosing regime. And the on-target bone marrow tox meant that efficacious exposures could only be reached in hematological cancers. So the aim was to develop a slow-release nanoparticle version with a more convenient dosing regime, something that could be dosed once weekly or less, perhaps had a higher safety profile that would then allow treatment of a broader range of tumors. And the technology that they chose was from a company called Bind Therapeutics. And here the drug is encapsulated in a polylactic acid matrix for controlled drug release. These matrices can be used in a variety of different modalities. And there's also inclusion of a PEG hydration shell, which protects from degradation, allows for extended circulation time of the moiety. And you can also, in these kind of constructs, include a tissue targeting molecule, although that wasn't part of this current program. So AZD1152 is actually a prodrug. It's the HPQA version that's the active moiety, actually. So here they use the HPQA moiety encapsulated in the PLA PEG nanoparticle, and that became known as AZD2811. And the preclinical studies did show that it delivered improved efficacy with a lower dose intensity and a reduced bone marrow tox. But generating these particles is not really off-the-shelf stuff, and they have to be refined and optimized to find the most suitable properties for the compound under study. And so development includes various rounds of optimization in terms of drug loading, release rates, particle size distributions, whole production process criteria that need to be met in order to deliver an efficient and a consistent product. And all these critical attributes that are changing as you optimize the product not only change performance, but also can potentially affect the safety as well. So the conclusion here was that despite all the existing knowledge, that the novelty of the new construct, the change in disposition of baracertib was sufficient to warrant further preclinical characterization. And so some new tox studies were done with the nanoparticle, which did in fact reveal some altered toxicity profile and some new target organ tissues, actually, which was well worth knowing prior to starting the phase 1 with this compound. So tolerability was good. There was a reduced bone marrow toxicity, and the compound's now progressing well through phase 2. The next example that I've got is one with Blueberry Therapeutics, and Blueberry are developing a new antifungal spray for the topical treatment of dermatophyte infections. And dermatophyte infections are a worldwide problem. It's high incidence, particularly in the West, things like athlete's foot, and it's an incidence that's already high and growing. And there are a number of treatments available. Terbinafine has been used for a long time, developed by Novartis. It's now over the counter. But still, there are some difficult to treat fungal infections. Onychomycosis is one of them, with the same dermatophyte that causes athlete's foot. This is where you get a really protracted infection beneath the nail and requiring oral tablets. And oral tablets are not necessarily a great solution here, because they do bring with them, you're obviously dosing the person systemically. It brings adverse safety profile with them, some target organ toxicity that you don't necessarily want. There are complications about drug-drug interactions. And so you're exposing the whole body rather than just the local infection. So the ambition here was to enhance drug penetration into the skin through the nail using a nanoparticle formulation, and this should allow significantly higher local exposures to be achieved using a topical administration route. So Blueberry had a state-of-the-art nanoformulation platform to develop here using drug cargo, in this case terbinafine, combined with a nanosyn polymer. And these things come together to spontaneously form a nanoparticle with a very consistent form, size, and structure. And also these nanoparticles have enhanced skin and nail penetration properties as well. So thinking about the safety then of this product, the two principal components, both been around for a very long time. I mentioned terbinafine had been around since the 1980s. Novartis had done a huge tox program by various routes to support their various creams, sprays, gels, tablets, etc. And so there's a huge wealth of clinical data already on the active moiety via both the topical and oral routes. However, much of the data was old. A lot of it wasn't to today's standard, and it wasn't easily accessible either. In terms of the nanosyn, this is a polyhexanide polymer. This has also been in widespread commercial use for at least 60 years. And there's a similar extensive database on the clinical safety of this agent. It's been used in wound dressings, wound irrigation solutions, contact lens solutions, swimming pool cleaners. It's used in things like lipsticks. But there's no prior history in pharmaceuticals. And again, although it had been sort of well profiled, the data was old and not really readily accessible. However, in this case, we came to the conclusion that given the extent of the human safety data and the concentrations that we were using in the local cream, actually further preclinical safety work would be of no real value in this case, really in informing the clinical safety profile of this product. So that was the argument that we made to the regulatory authorities. And to date, they've accepted that argument, and we've progressed into clinical trials in phase 2 now without doing further work. Although there was a lot of discussion as part of that with some back and forth, but it's currently successfully progressing through phase 2.
Rich, I'm going to ask you to go to your conclusion slide. I'm sorry.
No, that's okay. That was a third example. We don't need to worry too much about that. I think just in conclusion, I think, we've touched on this already. I think the principles that we can apply are consistent, whatever the modality. And obviously with complex medicines, there's a huge variety of different types of agents. But if we apply the same thinking and follow it through scientifically and build a package that makes sense from a scientific point of view, that's undoubtedly the right approach to take. So I'll leave it there then, Sarah.
Thank you, Richard. So I will just take this opportunity to thank Jenny, Juliana and Richard for 3 very full talks, such that we didn't get questions run on because you filled your time beautifully. I just want to introduce the speakers for next week. Next week will be our last in the series, and we're getting ready for the clinic. So we're going to hear from Claire Patterson from CEDAR about formulation, Emily Port from CPI around the scaling up, and Alan Boyd from Boyd's will tell us about how to navigate the regulatory landscape. So thanks once again for everyone joining us this week, and thanks again to the speakers. Okay, bye-bye.